So the LDL molecule, what does it do? VLDL transports the endogenous TG from the liver to the peripheral tissues along with the cholesterol. So during their course of the journey, what happens? The VLDL slowly liberates the TG contents towards the other tissues, so that the TG concentration of TG goes on decreasing, resulting in the conversion of VLDL to IDL. That is, the density, when the fat content decreases, the density increases. So very low density lipoprotein will be converted into IDL, right? And this IDL again further go on liberating the TG towards the skeletal tissues or skeletal muscles or adipose tissue or surrounding tissues, so that further again TG content goes on reducing, converting the IDL back to the ideal, then to the form of LDL. So low density lipoprotein. So VLDL, when converted to ideal, the density increases with the reduction in the TG content. Again, ideal when converted into the LDL, further the TG will be released. So majority, 90% of TG which was present in the VLDL is now out of the lipoprotein, so that it is the major concentration is now occupied by the cholesterol esters, right? So cholesterol. So LDL is rich in cholesterol. So what is happening in effect? VLDL transports the TG along with the cholesterol esters from the liver to the peripheral tissue. So during its conversion from VLDL to LDL, what is happening? The TG also will be liberated or will be coming out of the lipoproteins to the surrounding tissues or the peripheral tissues. Once the LDL is formed, LDL now takes up their function and it starts giving or distributing the cholesterol esters from the liver to the peripheral tissues. So what happens? The person becomes more and more fatty, right? That's why the LDL is known as bad cholesterol, right? And again, majority of the or 90% of the LDL is actually derived from the VLDL. So the main source of LDL is the VLDL itself. Only a remaining 10% of the LDL is actually synthesized by the liver cells, right? Otherwise, the majority of the LDL is synthesized from the, or is formed from the VLDL molecule itself, right? Only a very small part is, a very small concentration of LDL is actually synthesized by the liver cells, right? So that is happening. And once this and how does again the half-life, what is the time duration of an LDL molecule? The LDL, the half of LDL molecule is around 2 days. So by this 48 hours, it will be liberating most of its cholesterol esters from its structure, right? And this LDL again moves back to the liver cells, right? So how is it liberating, or how does it carry out its function? What is happening? What's the metabolism of LDL molecule? So LDL, again, when it reaches the peripheral tissues, it will be going and getting bound to the receptors because they are lipoproteins. So they cannot the lipid molecules cannot get directly attached onto the cells. So it is with the help of the apoproteins. So apoproteins is actually acting as a ligand. One of the functions of apoprotein is to carry the lipid molecules. Another function is that they are acting as receptors. I mean, they are acting as ligands for the receptors, right? So it is with the help of this apoproteins, the lipids are entering into the cytoplasm of the peripheral tissues. So the apoprotein, that is ApoB100, goes and binds onto the specific receptors present on the target tissues or the peripheral tissues, and the LDL molecule will be endocytosed. They will be taken into the cell, right? So that is how the metabolism is taking place. So LDL molecule will be taken up by the peripheral tissues with the help of receptor mediated endocytosis. That is the ApoB100 which is present on the LDL molecule. ApoB100 is the only apoprotein present in the LDL molecule, present on the surface of the LDL molecule. So with the help of this ApoB100, the LDL molecule goes and gets bound or gets attached onto the peripheral tissues. And once this ligand receptor complex is formed, the molecule will be taken, it will be endocytosed into the cell, into the peripheral tissue. And once it is in the cell, what is happening? The lysosomes comes and breaks the LDL molecule, right? LDL molecule, the lysosomes, and gets opened up, and the cholesterol esters will be liberated. That's how it is taking up the cholesterol. The peripheral tissues are taking up the cholesterol esters from the LDL molecule. Again, the LDL molecule or the peripheral tissues, the LDL molecule comes and binds onto the peripheral tissues, And there are specialized areas of the peripheral tissues upon which the LDL, the lipoprotein, comes and binds. These specialized regions that are present on the peripheral tissues are known as clathrin-coated pits. That is, there are certain specialized regions where the LDL molecule comes and bind, and those specialized areas we called as the clathrin coated pits. There are clathrin is a protein. So this protein in a protein clathrin a protein is there. So clathrin coat ചെയ്തിട്ടുള്ള കുറെ pit എന്ന് പറഞ്ഞ കുഴി. അപ്പൊ അങ്ങനെ clathrin coated pits ൽ മാത്രമേ the LDL the ApoB100 comes and binds. Let me see whether there is there are. So suppose this is a bloodstream. There is the this is yeah. The VLDL will be reducing in size to form ideal. Ideal acted upon by LPL to liberate TG and other apoproteins. Finally, the LDL molecule is formed. The LDL molecule will be having only ApoB100 on its surface, right? And this ApoB100 containing LDL molecule will be going and getting attached onto the peripheral tissues with the help of receptors, or through the receptors it's coming and getting attached onto the peripheral tissues. And on the peripheral tissues there are certain areas. So these receptors, LDL receptors, the receptors upon which the ApoB100 of the LDL binds will be specialized areas. There are certain special areas where the receptors can be found. അവിടെ മാത്രമേ LDL receptors കാണാൻ പറ്റുള്ളൂ. ആ regions അല്ലെങ്കിൽ ആ specialized regions ആണ് we called as clathrin coated pits. P-I-T, pit. So it's on the clathrin coated pits you can see the LDL receptors, and upon this LDL receptors this ApoB100 goes and binds. So as a result of the binding of the ApoB100 of the LDL molecule, the binding it onto this receptor, the receptor is present on the clathrin coated pits. The molecule will be getting fused and they will be taken into the peripheral tissues where it is acted upon by lysosomes, and the lysosomes and the lysosomes contains enzymes, right? So this enzymes breaks the LDL molecule, and what happens, the contents will be released out, right? So this is how the cholesterol molecules are coming out of the LDL molecule and is entering into the peripheral tissues. And once this LDL molecule are entering into the peripheral tissues, now the receptors are their function is over, so the receptors again go back onto this clathrin coated pit so that they can receive more and more amounts of LDL molecules. Clear? So this is how the metabolism of LDL happens. And the lysosomes contain different types of enzymes. Lysozymes, we call it as lysozymes. So what are the functions of lysozymes? The lysozymes breaks up the LDL molecule and liberates the contents in the LDL molecule, and the contents present in the LDL molecule is mainly the free cholesterol esters. Cholesterol esters will be liberated and be getting deposited in the peripheral tissues, right? So again, the majority of the LDL molecule will be degraded, and once this cholesterol contents are released, they will be again going back to they will be coming out of the peripheral tissues, they will be released into circulation, they will be taken into the liver. And so 70% of the LDL molecule will be degraded. The catabolism or the breakdown will be happening. The destruction, that is the removal of ApoB100, will be taken back in the liver itself, right? And only very few quantities will be happening of LDL molecule will be undergoing breakdown in the extrahepatic tissues. So this is how the metabolism of LDL take place. And what are the functions of LDL? The LDL, the majority of the plasma cholesterol transportation ആണ് LDL function. അതേസമയം VLDL function TG transportation. ഏത് TG? Endogenous TG. While chylomicrons function transport of exogenous TG. Clear? So each lipoproteins has got different functions to be carried out. Chylomicrons transport the dietary TG, the dietary cholesterol. Diet derive ചെയ്തിട്ടുള്ള ഏത് lipid molecules അല്ലെങ്കിൽ lipid related ആയിട്ടുള്ള compounds transportation is carried out by chylomicrons. While VLDL transport the endogenous TG, right? And the LDL transports the cholesterol esters. Clear? To the from the liver to the peripheral tissues. So VLDL transports the endogenous TG from the liver to the peripheral tissues, right? So the main function of the LDL molecule is to carry out the plasma cholesterol. Majority cholesterol transportation from the liver to the peripheral tissues is carried out by the LDL molecule. Then it's used അവിടെ ചെന്ന് കഴിഞ്ഞാൽ cholesterol function, what's the function of cholesterol we are going to study in the next portion. We are coming into the detail of the cholesterol metabolism. But cholesterol, tissue deposit cholesterol function in the cholesterol will be used for the synthesis of steroid hormones, the bile acids, etc. Or it will be deposited in the membrane so that they can shield or protect the person. Then they can be used for the esterification with MUFA, monounsaturated fatty acids, right, etc. So several functions are there for cholesterol molecule once they are getting deposited in the peripheral tissues. So this is how the metabolism of LDL takes place in the body. Now another one is, so LDL ഒരു bad cholesterol ആണ്, while because it is an atherogenic molecule, that is it can induce the thrombus formation, blood clot, blood vessels deposition, fat deposit we call this thrombus formation. So LDL molecule can undergo different type of modification. That is, LDL can be subjected for oxidation നടക്കാം, അല്ലെങ്കിൽ glycation നടക്കാം. അപ്പോൾ അങ്ങനെ modified substances ആകുമ്പോൾ these modified LDL will be acted upon by various types of molecules like hydrogen peroxide, nitric oxide, and different superoxides, right? The LDL molecule will be attacked by different types of molecules, so that LDL undergoes conversion to give like oxidized product or glycated products and all. So what happens? These oxidized LDL molecule will be immediately attacked by the body's immune system, immune cells, that is macrophages, monocytes, etc. will be attacking these modified LDL molecule, and they will be taken into the target site and they will be subject for destruction. So this LDL, which is carrying the cholesterol molecule, start getting deposited in the blood vessels, right? When more and more fat remove ചെയ്യാൻ പറ്റില്ല, so they will be forming plaques. They will be forming they will be getting sticking onto the capillary walls. Up in the capillary walls the size അവിടെ കുറയാൻ തുടങ്ങും. So blood flow will be reduced, which results in atherosclerosis or atherogenesis. അതുകൊണ്ടാണ് myocardial infarction വരാൻ കാരണം LDL molecule, because LDL will be undergoing modification. It will be converted into glycated products or oxidized product. It will be immediately recognized by the immune system. Because it's a normal substance. LDL, okay, it's a normal substance. But LDL, when undergoes modification, will be converted into oxidized LDL or glycated LDL. So it will be immediately recognized by the body's immune cells. And these immune cells will be taking up the LDL molecules towards it, and they will be getting deposited on the walls of the capillaries, so that the smooth blood flow will be affected, and there will be a hindrance in the smooth flow of the blood, which results in the atherosclerosis, plaque formation, and myocardial infarction. That's why it is known as bad cholesterol, right? So it's actually the immunological aspect. You just see that it's monocytes, macrophages, etc. So this is the LDL molecule. The LDL, this is a LDL molecule which is carrying the cholesterol esters. See, the cholesterol is transported by the LDL from the liver to the peripheral tissues. So during the course of the journey, what is happening? The LDL will be acted upon by reactive oxygen species, that is like hydrogen peroxide, superoxide, free radicals, etc. So converting the normal LDL to modified LDL. LDL undergoes some sort of modifications. So this modified LDL will be immediately recognized by the body's immune cells. When macrophages, monocytes recognize it, what happens? They will be taken up and will be subject for digestion. Actually destruct but what happens? These macrophages and LDL start getting deposited on the walls of the capillaries. Thrombus formation, form cells, thrombus. Blood so this area is reduced. It is occupied by the fat molecules, so resulting in atherosclerosis. So that's a defect of, or that's a disadvantage of increased values of LDL in the body, or that's why it is known as an atherogenic substance, right? So that is the LDL. Then lipoprotein A is there. Again, we have already discussed. It is a very dangerous substance that is found in the body, and actually lipoprotein A is a modified form, because it is lipoprotein A is having an additional apoprotein, that is that's why it is known as a little rascal, because the lipoprotein A contains ApoA. A is the small letter and is always found to be in linkage with ApoB100, right? So ApoB100 disulfide linkages. So it can again inhibit the plasminogens, causing thrombus formation.